So objective of our time together now is to understand the importance of cell membranes. See, the evolution of the cell membrane must have happened much, much earlier than the prokaryotic cell, right? So life as we know it is existing because of the evolution of the cell membrane. And here's a animation of a eukaryotic cell membrane, like a cell membrane that's found in you and me. So you can see the phospholipid bilayer, and you can see how busy it is. It's studded with receptors, and there's molecules moving across. So this is a huge discovery, the structure of the cell membrane. And we are finding new things every day. So here's a quick flow chart. So the main focus would be how molecules will move across the membrane. Sometimes it's passive. That means it doesn't require energy. And sometimes it's active. That means it requires energy. Under passive, we have two basic kinds. It can either be through diffusion, it's called simple diffusion, or a process called osmosis. And under simple diffusion, there's a few types. Under active transport, we will have the pumps, like we'll look at the sodium-potassium pump, or molecules can move via a vesicle. If it's coming in, it is called endocytosis, inside the cell. If it is going out, it is called exocytosis. So what is the function of a cell membrane? So the cell membrane, it is the final barrier between the cell and the environment. So it plays a huge role in maintaining homeostasis in a cell. So apart from giving the cell structure, maintaining the integrity of the cell, it also controls transport of materials inside the cell. So they will call it selectively permeable. So it's going to regulate the molecules that's coming in and out of the cell, all to maintain homeostasis. It will also play a role in cell-to-cell communication, because one cell is not like living by itself, except if it's a prokaryotic bacteria, it can live as a single cell unicellular organism. But complicated eukaryotes are constantly in a tissue, the cells are like communicating with each other. So that's the function of the cell. So apart from protection, it's also maintaining homeostasis and communication. And then it will also form these gradients that I'll talk about, which is very important. Here's a nice animation of how the cell membranes play a vital role in a cell. So why is it important to study the cell membrane? I just want to give like a few examples. There are many, many diseases known to human beings, like cystic fibrosis is something going wrong with the receptor that is found in a cell membrane. Receptor is nothing but a protein, that has a vital function of transporting molecules. And when one of the receptor proteins here in the membrane in the respiratory system is blocked, there's an accumulation of mucus. And that's a deadly disease called cystic fibrosis. And then there's cystinuria, the transport of materials, especially cysteine, between blood and urine is blocked. And then all these, huge crystals forming in the kidneys, thereby blocking urination. It's a bad disease. And there was a movie recently, peroxisomes that control, they are full of peroxide, and they play a role in hydrogen peroxide metabolism in the body. So this movie is a real story about a boy. He has a deficiency of a membrane protein in the peroxisome. And because of this, you can see there's all this accumulation of plaque in his brain. So they made a movie out of it. If you get a chance, watch that movie. We have to understand cell membranes very well, because this is how we target drugs for controlling microbes. When you take an antibody, it's not going to affect our cells, but it's going to kill the bacteria that's in the body. So you have to know what is the difference between a bacterial cell wall and our cell membrane in order to make effective drugs. And here's, like diabetes has become so common. People don't think it's serious, but it's like an epidemic because of bad lifestyle choices and because of the food that we eat. People are being diagnosed with diabetics so frequently nowadays. And again, it's because of a malfunction of the insulin receptor in the cell membrane. There's different types of diabetes, but this is a picture where in diabetes 2, there's something called insulin resistance. And again, it's going back to the cell membrane. But like more relative is here we are like in the middle of a pandemic. How does the COVID-19 virus enter a cell? How come it's escaping the immune system when it attacks a cell? It's because it's able to trick our cell receptor. So see, there is a receptor in our cell membrane called ACE2 receptor, ACE2. And then our cell unknowingly thinks COVID is a friend, and then it brings the COVID virus into the cell. So now you can see the importance of cell membrane. So this is how the enemy is getting into the cell. This has profound importance. Among all the structures we have studied in the cells, cell membranes are super important. So fluid mosaic model of cell membranes, I already explained in our last lecture. But basically, why is it called fluid? It is made up of a phospholipid bilayer. So this is the structure of a phospholipid. So there are two layers of it. So it's called phospholipid bilayer. It is called fluid because it's not static. There's a constant, they're all moving. Again, it's so amazing to think the dynamic nature of cell membranes. The person who deciphered the structure of cell membranes, he got a Nobel Prize. Huge discovery. So all these molecules are floating around. And then it's called mosaic because there's all these molecules of proteins and cholesterol and lipids that are attached to it. See, some proteins are going right through. So it's called integral protein. Some proteins are attached to the surface. They are called peripheral protein. And you can see some cholesterol molecules, very important for the fluidity of the membrane and to maintain the integrity of the membrane in like high temperatures and cold temperatures. And here you can see something called glycoprotein. So glyco means sugar that is attached to a protein. So they will play a huge role in like antigen antibody reactions. Here's a glycolipid. That means glyco means a sugar that is attached to a lipid. So this will play a role in cell-to-cell communication. So you can see that the cell membrane is a very, very busy place. The nature of cell membrane, remember this, is selectively permeable. It is going to, it's like the security guard of the cell. It's going to allow some substances to enter the cell and it would stop some substances from entering the cell. So you need a ticket to get into the cell. And here, if you zoom into the structure of a phospholipid, it's an amphipathic molecule. It has a hydrophilic head, a polar head, and a non-polar or hydrophobic tail. So here, right in the center, it is hydrophobic. So the heads will face towards the outside of the cell, which is an aqueous environment. That means a water-based environment. And in the inside of the cell is also aqueous. It's all water-based. So all the hydrophobic heads will line up like this. So if the molecule is small and non-polar, like oxygen, carbon dioxide, they're just going to like wiggle their way inside the cell membrane. Very easy for them because they're so small and they're not charged. Non-polar molecules. Whereas if they are uncharged polar molecules like water and glycerol, they would need help to get into the cell. So this is like a permeability scale that you're seeing, highly permeable to low permeability. See, glucose and sucrose are big molecules. they need help. They might need a protein in the membrane to escort them inside the cell because they are large and uncharged polar molecule. Ions like sodium, potassium, chloride, they'll all need help. So depending on the size and charge of the molecule, the permeability is differing. So this leads us into the phenomena of osmosis and diffusion as to how molecules will enter the cell. So when you say diffusion, it does not use energy. So it is called passive transport. So diffusion is the movement of molecules from high concentration to low concentration along the concentration gradient. So this is because of the inherent kinetic energy of the particles. So there's like Brownian movement in the particles. The movements are always shaking. So when there is a concentration gradient, they're going to just move from high concentration to low concentration. A very good example of this is our breath right now. See, right now, you and I are breathing. So oxygen is just moving from the air that you inhaled into the blood because the oxygen content inside the blood is low. So oxygen is just diffusing into the blood, and carbon dioxide, which is a waste because of metabolism in our body, is going to diffuse out. So right now, diffusion is happening in your lungs. So these structures are called alveoli of lungs. This is where the gas exchange happens. So excellent example of diffusion is oxygen going into the blood and carbon dioxide leaving the blood in the respiratory system. So if somebody is wearing perfume, you know it, right? Because the perfume is diffusing from their clothes to the air and you can pick it up. So examples of diffusion is all around us. There are 3 types of diffusion. So here, if it is like oxygen and carbon dioxide, they're small lipid-soluble solutes, molecules, so they just wiggle their way into the membrane. But if certain molecules would need help. So here's a glucose molecule. It is using a protein. So the protein is grabbing it from outside the cell and pushing it inside the cell. So this, because it's facilitated by a protein, it is called as facilitated diffusion. If there is a protein channel, right? It's called as a channel protein. So it is channel mediated facilitated diffusion. For example, water will use a protein called aquaporin to escort it to enter the cell. So again, no energy is required. And this is happening constantly across our cell membranes. So when we talk about the movement of water from high concentration to low concentration, then it's called osmosis. So this is the famous, I've seen this picture as a U tube, or here you can see these are 2 different solutions. The red and the white ball is talking about water molecules. So there's more water here compared to the other side. So there's more water on the left side. So water is going to move from high concentration to low concentration. And you can see how the water level is rising up on the other side. So the movement of water across a selectively permeable membrane is called as osmosis. Again, it does not use ATP. An excellent example would be how water enters a plant. So when you water a plant, there's more water in this dirt. So what happens is water is moving from high concentration to low concentration inside the cell. So remember, osmosis also does not require energy. So the next concept you have to understand is tonicity. So every cell can be in different strength solutions. So what happens? So like when you say solution, it means it has a solvent and a solute. Sugar solution. Water is the solvent and sugar is the solute. So plant cells and animal cells will differ in their response to different tonicities. So here's an animal cell. So this is a blood cell. So you can see if you put the blood in an isotonic solution, that means the strength of the solution inside the cell and outside the cell is the same. So the amount of water entering the cell and leaving the cell is same. So the cells are happy here. Whereas if you put animal cells in a hypertonic solution, that means the strength of the solution is very, very high. And the water content inside the cell is high compared to the outside. So what will happen is, if you put it in a hypertonic solution, it will lose water and shrivel up. So this is a bad state to be in. You don't want that. Whereas if you put it in a hypotonic solution, that means the water content is high outside the cell. So water will move from outside to inside the cell. The cell will become too big and it will burst. So for animal cell, isotonic condition is the most ideal. Whereas plant cells, they differ because they have a cell wall. So when you drop when a plant is subjected to a hypertonic situation, it will lose water and it will wilt. In isotonic, what's going in and out is the same. But however, plants kind of prefer a hypotonic condition so that the vacuole is nice and filled, so that it's nice and turgid. So they'll call this the turgid. This phenomena is called plasmolysis. So now why is all this important? See, if you've been to the hospital, right? Let's say you are sick and they want to inject or like give you some saline solution. You know, if the saline was hypertonic, your blood cells will shrivel up. If it was hypotonic, it's going to burst. So the saline has to be an isotonic solution. So again, there's this tragedy that happened in a hospital where when they used to mix saline in the hospitals, but now it's computerized, it comes in bags. So they gave the wrong tonicity and then there was a loss of life. So let's talk about active transport. This is super important because if it is your neuron, right? If you're talking about your brain cell, it is when you talk about a nerve impulse or the action potential as it's called, it's all because of this proton, pump, sodium potassium pump. So here in the cell membrane, we have a protein. So ideally, we want to have more sodium outside the cell. So what the cell will do is it will pump out the sodium and then it will keep the potassium inside in order to maintain the action potential or the electric current over the cell. So for this to happen, it will use ATP. So the picture in the bottom left, you can see ATP is used to pump 3 molecules of sodium outside, and it will bring in 2 molecules of potash on the inside. So there'll be, it is maintaining the right gradient for homeostasis to happen. So this is called sodium potassium pump. And this is what a nerve impulse is. I know this is a little sophisticated for you to understand now, but know that ATP is required for active transport. So here we are moving molecules against the gradient, not along the gradient. And then this will trigger neurotransmission and so forth. I want you all to see this picture because I'm going to ask you a question later. You know, this is a neuron. So this is what your brain is made up of. This is the nerve cells or neurons that can be found in your brain. And then when one neuron fires, it will release a neurotransmitter. And then that neurotransmitter will trigger the next neuron. It's amazing. All right, those are all molecules. What if a cell has to bring in a lot of stuff in? So what it will do is, it's easy. It's called endocytosis. So here's a large particle of food that a cell wants to bring inside. So the cell membrane will pinch it off. It will invaginate. It will pinch it and form a vesicle and bring it inside. So if it is a solid, it's called phagocytosis. If it is a liquid, it's called pinocytosis. Cellular eating, cellular drinking. But let's say a receptor. Receptor means a protein in the cell membrane that is going to have a specific attachment to a molecule. So if a receptor is involved, it's called receptor-mediated endocytosis. So here you can see a white blood cell is actually chasing a bacteria. And then it is going to engulf the bacteria using phagocytosis and destroy it. Isn't that a nice animation there? So to summarize, what we studied was the structure of membranes is super important. Understand its structure. Understand it's the fluid mosaic model, that it's very, very dynamic. It is selectively permeable. It is going to allow some substances, and then some substances will need proteins to enter the cell, like a carrier protein, channel protein. So in active transport, if it is going against the concentration gradient, it will require ATP energy. Passive transport is movement from high concentration to low concentration. No energy is required. And examples we saw was diffusion and osmosis. It's like going down the river on a tube. You know, you're not paddling, nothing. You're just going with the flow. Whereas active transport, for me in my mind, A stands for ATP. Active transport requires a lot of energy because you're going against the current, against the concentration gradient. You're moving from low solute concentration to high. Energy is required. And good examples we saw was the sodium-potassium pump, endocytosis, and exocytosis. And I hope that was helpful for you.